looks like everyone's ready to go. So our next speaker is Dr. Geraldine Allen. She's coming to us from the University of Victoria, and her presentation will be Native and Introduced Phragmites australis in British Columbia. Right. Thank you so much. Take it away.
All right. Thank you very much. Well, thanks very much for inviting me to this conference. And I'm actually working closely with Becky Brown, who just talked in the other session. And Becky is an invasive plant specialist for the British Columbia government. And Becky and I have been working for about the last 10 years, over 10 years now, on identifying native and non-native Phragmites in Western Canada. And the reason that's an emphasis for us is that we have a kind of different order of problem than people do in the Eastern Canada. I'm just going to start with a bit of an overview here. This is a worldwide map of known localities for Phragmites australis taken from the GBIF website. And so you can see it's extremely common in Europe and in North America, and it's a worldwide grass that occurs in temperate and tropical wetlands all over the world. And so this map certainly is an underestimate of its presence in many parts of the world, but it certainly gives a glimpse of how it is in North America and Europe. And as you all know, Phragmites wasn't so common in North America. It wasn't a pest yet, maybe 150 years ago. And in the last 150 years, it has spread rapidly right across the continent, starting from its introduction in Eastern North America. And Kristen Saltonstall, she's now a diversity ecologist with the Smithsonian Tropical Research Institute, but back in 2002, she was doing her doctorate at Yale, and she developed DNA markers to tell the native and non-native subspecies apart. And if you don't have those, they're actually very similar, and I'm sure many of you have encountered this. They're pretty hard to tell apart unless you have certain kinds of structures. And so it's easy to have a lot of plants which you're not sure are native or non-native. Now, this shows the work that she did where she looked at old and new samples of this species in herbarium. And she took samples of herbarium specimens and showed that if she looked at old specimens, they were mostly of genetic, the markers suggested genetic forms that were all native, and only a few on the eastern coast of North America were non-native. And then as she looked at later ones, you could see how the what she called haplotype M, which was the chloroplast marker variant that was the invasive form, was spreading right across the country. And this, of course, is based on the US, but something similar was happening in Canada. Now, the non-native subspecies has continued to spread all over North America, but it's still much more common in the East than it is in the West. In the West, we have scattered populations of it in BC, and in a similar situation in the Western US. It's often found in locally wet areas, riparian areas along the coast, but it's not nearly as common in the average habitats. Maybe just wetlands are just not as common. This is a map showing the sampling that Becky did with her group, looking at occurrences of Phragmites all over British Columbia. And she collected samples for us, and what we did is we applied those DNA markers to see which ones were native DNA variants and which ones were non-native. And the red dots represent the introduced subspecies australis, and the green dots subspecies americanus. And as you can see, even now, most of the samples in British Columbia are still the native variant. So if you are to intelligently direct invasive species control measures, you have to know what you're looking at and which populations are native and which ones are not. Now, this is a this network diagram shows all of the genetic variants, the chloroplast haplotypes that were known worldwide in 2017 when we published a paper on this. And so any little circle with a letter inside it is a known variant, a known haplotype, and the dots, the black dots, just show how many differences there are between one haplotype and the next. So you can see there's a cluster on the lower part of this network has quite a few different variants, and those are the ones that are native to Europe and Asia. And the ones at the top right are the variants that have been found in Western North America, in North America overall. And the colored dots show the ones that we found in our study. So these are ones known from British Columbia or in Western Canada. We found two variants, L and M, that were non-native, and then we found 4 that were native. And this is, this is how they're distributed. They plot on the left, the map on the left shows the distribution of non-native variants. And we found 2 different non-native variants. Most of the non-native genetic forms in North America all belong to this M group, which is what you might expect if they had arrived in more or less one region of the continent and then spread across from that initial introduction. There have been recent records of more introductions. So L represents another variant that's turned up in BC, and it's been found here or there. The first one we found in BC on southern Vancouver Island there actually turned out to be a variegated form, which was a cultivated plant, but it's also, it's in the invasive forms that are around. And on the right, there's the map shows all the different native variants that we found amongst the samples that we looked at. And most of the samples that we looked at were native. We analyzed around 270 individual plant samples, and of those, only about 20 of them were non-native. So most of the localities in BC actually are the native form of Phragmites, and only about 10 or 12 localities have the invasive form. And so this is very important information for Becky so that she knows where to direct your efforts and how to follow the advice that other people have given of getting on top of any problem very early on and before it becomes, well, it's still a small problem before it becomes a big one. So in BC overall, Phragmites is widespread but sporadic. And the question is it still spreading? It is probably still spreading, but the occurrences, as I said, are relatively few. And the known localities are only about a dozen, and those are being very carefully managed. And BC has a rapid response program to really keep track of these ones. Now, why is this species so much more common in Eastern North America than Western North America? And should we be worrying that one day we'll have it everywhere and we're going to be doing some of the things that are being done in Ontario? This is a paper that came out a few years ago, which used climate modeling approaches to predict where the known genetic variant that has been introduced into North America from Europe, where that could be expected to be most common. So this is a climate model for the M haplotype. So the way this is done is that known localities are identified, and then the climate variables from those localities are used to build a predictive model to see where else in the world that climate might exist, and therefore where else that variant might do well. So the blue dots represent the native range, and the red dots represent the introduced range of the haplotype M, the invasive variant that we have in North America. And you can see what the model shows brightest colors, red especially, shows high probability of doing well in an area. And you can see that it predicts it for Europe, where it is indeed common. It predicts it for Eastern North America, where it's also common. This is a close-up of the North American map. And what it essentially predicts is that habitat on the whole is more suitable for it in Eastern North America than it is in the West. There certainly are areas it's predicted in the West, but nothing as extensive as in the East. So it's much more localized. In Western North America, we see native populations in many habitats. Of course, it's a wetland species, so its occurrences are scattered even in the West. Excuse me. But the invasive populations are established particularly in estuarine habitats, along lake margins, in desert washes with intermittent water. And the favorable habitat is not continuous, so that's advantageous for controlling spread. But nevertheless, there are areas where it can grow, and those are susceptible to establishment of the non-native form. So one of the things that we were doing, as well as looking at the distributions of native and invasive forms, was to see whether we could collect additional data on how to tell these forms apart using just morphology. This has been looked at. It was looked at by Dr. Saltonstall and by others who have tried to find ways of telling the two subspecies apart that doesn't necessarily involve sequencing DNA regions. And so we took the samples for which we had looked at DNA, and so we knew what the DNA said about what subspecies they were, and we looked at morphological differences between those groups. And these differences I have here were already known. We already knew the native subspecies is smaller, it has narrower leaves, less dense inflorescence, and it has reddish stems, whereas you don't see those typically in the introduced subspecies. And the details of the flowers, the floret structures, the bracts associated with the flowers are larger in the native species, but smaller in the introduced one, although the non-native has a lot more flowers in a flower head. And the ligule, which is a little paper-like structure where the leaf sheath and the leaf blade meet, is different as well. But no one of these really separates the two subspecies. There's a bit of overlap, even though they're average size differences. So we were interested in whether it would be possible to combine these in different ways to see if we could make an index that would separate them. So we looked at the glooms, which are the flower bracts, the outer flower bracts, and the lemmas, which are the inner flower bracts, and the ligule of the leaves. And you can see that in all cases there are differences, but there's a lot of overlap. We took 11 different traits and we did a multivariate analysis, which is shown on the right, which shows that if you have information about all those 11 traits, there's pretty good separation between native and non-native subspecies. But that's not as convenient for actual on-the-ground separation. And we developed a couple of indices which are more useful, in which we assigned a scale value to each of the characters we were looking at, and then we added up the values to get an overall scale from 0 to 10, and then we put the native and non-native forms on that scale. And we found that either a 2-character or a 3-character index could separate the plants in our study. Now, the limitation of that is that we only use plants from BC, and we only use the couple of hundred plants that we had information about. And so we don't know how well that applies to elsewhere in North America. Now, another thing we were looking at was the possibility of hybridization. And there's not very much information about that. It's been reported that the two subspecies can hybridize. And a worry about that is that even where the prevailing subspecies is the native one, subspecies americanus, if they hybridize, you could get invasive genes going from one subspecies to the other. They spread vegetatively mostly, but hybrids have been documented. And so we're interested in that. This is a study done in the Colorado drainage basin, in which hybrids were reported. And the diagram at the top shows molecular data, in which the dots in the middle are the hybrids, and they were making established clusters, established stands in between native and non-native populations. This is just in a small area, presumably still small enough to control, but it's something that one needs to keep in mind where the two subspecies occur. So we did most recently, we looked at some samples in which we looked at a DNA region in the nuclear genome, the ITS region, which is very commonly used to look at variation within species. It shows a lot of among individual variation. And so we were interested in whether comparing the chloroplast and the DNA and the nuclear DNA markers would indicate hybridization, because hybrid plants could often carry markers in which they inherited one thing from one parent and one thing from another. There are only a couple of localities in BC where both of these subspecies are known to occur together. In the south of the province, in the Creston Valley, and also in the South Okanagan, where there's a lot of Phragmites, and both native and introduced subspecies occur. And we didn't find any hybrids, but as a result of this, what we did find is that the ITS DNA region is also really good for separating out the native and non-native subspecies. So it's an additional DNA marker region, which hasn't been much used, but it's just about as easy to sequence as the markers proposed by Christian Saltonstall. And so it's another way of telling these things apart. So if you have a leaf fragment and you have no idea what subspecies it is, it's possible to take just a tiny little piece of that and get DNA out of it and readily figure out what subspecies it is. And we've been doing some of that for Becky. Now I just want to finish up here by thinking about what the future holds for the control activities in the West. It looks from the climate modeling as though in Western North America, the invasive form of Phragmites is just more limited by climate. It's a summer dry climate in large areas of Western North America, in contrast to the east, where the summer is often wet and rainy, and evidently Phragmites likes that eastern climate and not so much the western climate. So Phragmites tends to only occur in the West in areas where it can have moisture around its roots all year. And so that's usually along estuaries along the coast, or in riparian habitats, lakes and streams, which offer places where it can establish. And those are the habitats we see in British Columbia. There are lake river systems along the Columbia River in the Okanagan Valley, where there's large areas of wetlands. And that's where we have the most problem with Phragmites in the West. And we haven't found any hybrids so far. And so, and maybe we won't, because the introduced variants are being closely monitored, but they are being found more frequently overall. And we see from recent studies in different parts of North America that new variants of Phragmites that are not native to the continent keep being introduced. There's some new ones reported quite recently in California that are different from the M haplotype, which is otherwise everywhere. And so not only keeping track of hybrids, but just keeping track of new variants, especially where the two subspecies co-occur, is an important thing to do.
2 minutes remaining. 2 minutes remaining.
I'm just about done. So here I just want to wrap up by acknowledging the help that I've gotten. I have been doing all this DNA sequencing in my lab at the University of Victoria, but all the specimens came from Becky's group, and this is a long list of all her co-workers and summer field assistants who have supplied us with samples. And we've also based some of our work on herbarium specimens that we got from the Royal British Columbia Museum. And the funding was provided by Becky's group, the Ministry of Forests from the province of British Columbia. And that's all. Thank you very much.